All right, welcome to Rad Onc Smart Review, RadBio Edition.
Hey, everyone.
Today, we are jumping straight into one of the absolute core topics for any radiation oncologist, really how cells fundamentally deal with the damage we inflict with radiation.
Exactly. Think of this as dissecting the cell's really intricate emergency response system, the DNA damage response, or DDR.
Right, and specifically today, we're focusing on how cells first sense those, nasty double-strand breaks and replication stress.
And the absolutely critical roles of three key players, ATM, ATR, and γH2AX. You'll hear these a lot.
Yeah, understanding this molecular signaling network isn't just about checking a box for your board exams, although it's definitely key for that.
Definitely. But it's vital for appreciating why radiation works, why sometimes it doesn't, and crucially, why some patients are incredibly sensitive to it.
So we're going to walk through the sensor proteins, the signal transducers, the effectors, how they coordinate cell cycle checkpoints.
And connect it all back to the clinical relevance you really need to know cold. This is like foundational stuff for mastering your RadBio boards. Seriously important.
Okay, so let's get started maybe by asking the fundamental why. Why is this DNA damage response system so utterly crucial in radiation biology?
Well, because ionizing radiation, first and foremost, it causes DNA damage. That's its main effect. And among all the different types, DNA double-strand breaks DSBs, they're the most lethal for the cell.
Right, the most dangerous lesion.
Yeah. If a cell doesn't sense and repair those correctly, it's either, lights out for the cell through apoptosis or maybe worse.
Worse.
Well, it survives, but with mutations and genomic instability, and that's how you can get cancer developing down the line.
Got it. So maintaining genomic integrity is like the cell's top priority for survival and, preventing malignancy.
Exactly. And the DDR is the cell's main defense mechanism. It's a really coordinated effort. Sense the damage, blast out the alarm signal, recruit the repair crews.
And importantly, slam the brakes on the cell cycle. Got to buy time for repairs.
Right. Or even trigger programmed cell death, apoptosis, if the damage is just overwhelming. It's the difference between a cell recovering, dying, or, recovering incorrectly.
Yeah. Absolutely
fundamental.
Okay, so how does the cell even know it's been damaged? Especially by something like a double-strand break. Who are the first responders on the scene?
This is where we meet the, commanding officers of the DDR army. Two master kinases right at the very top of the signaling cascade.
And those would be ATM and ATR.
Correct. ATM, which stands for ataxia-telangiectasia, mutated a mouthful, just remember ATM, and ATR, which is ATM and Rad3 related.
Okay.
They are both members of the PIK family. These are large kinases involved in sensing and responding to DNA damage. They act as the primary sensors and signal transducers. Big job.
Okay, so ATM.
Yeah.
That's our main guy for double-strand breaks, which, like we just said, is the signature damage from X-rays and gamma rays, right?
Exactly. In a healthy, undamaged cell, ATM is just kind of hanging out, usually inactive. Often it's in a dimer or maybe a multi-Mortier form.
Just chilling.
Pretty much. But the second a DSB occurs, another complex, the MRN complex, is on it instantly, zero delay.
The MRN complex. Okay, tell us about that one. What's in it?
So MRN is like the initial reconnaissance team. It's made of three proteins, MR11, Rad50, and MBS1.
MR11, Rad50, MBS1.
Got it. Yeah. MR11 has nucleus activity, so it does some initial processing at the break ends. Rad50 uses AT-pace activity, basically uses energy to kind of hold the two broken DNA ends together, bridge the gap.
Okay.
And NBS1 is the critical piece. It senses the break and directly recruits that inactive ATM to the site.
So MRN grabs onto the brake, holds it, tidies it up a bit, and then basically yells, "Hey, ATM, get over here."
That's a pretty good analogy, yeah. Once ATM is recruited by NBS1 to the DSB, it gets activated. A key step is ATM phosphorylating itself.
Autophosphorylation.
Right, at a specific site like serine 1981 in us mammals. That phosphorylation causes the ATM dimers or multimers to split apart into active single units, monomers.
And boom, active kinase.
Exactly. Full kinase activity unleashed. And ATM is primarily responsible for responding to these DSBs in G1, S, and G2 phases of the cell cycle. Covers most of it.
Okay, so ATM called by NBS1 to DSB, autophosphorylates at serine 1981, becomes active dimer or multimer works, is gone, S and GT but a woo. What about ATR?
Okay, so ATR is the sensor for a different kind of problem. Single-stranded DNA or ssDNA.
Which isn't usually the direct result of radiation, right?
Not usually the primary damage, no. But ssDNA arises from other types of damage or just normal cellular processes that go wrong. The classic example is stalled replication forks. That's a huge source of ssDNA stress.
Like when the DNA copying machinery gets stuck.
Exactly. Or when the cell is processing UV damage, that can create ssDNA. Or even during DSB repair itself, specifically the end reception step that happens in homologous recombination repair that generates ssDNA tails.
Okay, so if replication gets jammed up or DNA gets unwound for certain repairs, ATR steps in.
That's the idea. ATR works very closely with its essential partner protein, ATRIP. ATRIP stands for ATR interacting protein.
Makes sense.
So when ssDNA gets exposed, another protein called RPA replication protein A quickly coats it. This protects the fragile ssDNA and keeps it extended.
Okay, RPA coats the ssDNA.
And then ATRIP specifically binds to this RPA-coated ssDNA. And that interaction is what recruits ATR to the site of the problem.
So RPA coats the ssDNA, ATRIP sticks to the RPA, and then ATR comes along with ATRIP. Kind of a chain reaction.
Yep, that's the core of it. And there are other proteins involved too, like top BP1 and the 911 complex.
The 911 complex, like the emergency number.
Yeah, easy way to remember it. It's made of Rad9, Rad1, and Hus1. It actually forms a clamp-like structure around the DNA, similar to PCNA in replication, and helps get ATR fully activated.
Gotcha.
So ATR is absolutely essential for the S-phase checkpoint when replication is happening, and it also plays a major role in the G2M checkpoint, sometimes taking over from ATM later in G2.
Okay, so quick memory tip here, really important for the boards. ATM is primarily for double-strand breaks, think radiation damage.
Right.
ATR is primarily for single-strand DNA and replication stress, think stalled forks, UV damage processing, that kind of thing.
Exactly. And previewing a bit, remember this pairing. ATM primarily signals through the kinase CHK2. And ATR primarily signals through CHK1.
Okay, ATR CHK1 and ATM CHK2. Got it.
That ATM CHK2, ATR CHK1 pairing is super high yield. You'll definitely see questions testing that.
Okay, so ATM or ATR get activated right there at the site of damage. What's one of the very first things they do, especially ATM at a DSB? How does the cell mark the spot, like put up a flag so all the other repair proteins know where to go?
Yeah, this is one of the most fascinating and widely studied early signals. It's a modification that happens to the chromatin itself right around the break.
To the histone protein?
Exactly. One of the initial widespread modifications catalyzed by ATM and also ATR and another kinase called DNA-PK, which is involved in NHEJ repair, is the phosphorylation of a specific histone variant called H2AX.
And that phosphorylated version is gamma H2AX.
Precisely. They slap a phosphate group onto a particular serine residue, serine 139 in mammals. This modified H2AX is now called gamma H2AX.
Okay, so think of gamma H2AX as the cell literally painting a big, bright distress signal onto the chromosomes right where the damage occurred, like putting up a flare at an accident site.
Exactly like that, but it's more than just a marker, right? It's not just passively sitting there.
Definitely not. This phosphorylated H2AX creates a specific modified chromatin environment, essentially a landing pad or a scaffold, and it spreads out, sometimes for megabases, huge regions around the double-strand break.
A landing pad. I like that.
Yeah, this altered chromatin landscape becomes a crucial platform for recruiting a whole cascade of other DDR proteins. It organizes the response. Okay, so it's not just a flag. It's more like a construction site where all the other workers are told exactly where to gather and what to do.
Perfect analogy. And a key protein that binds directly to this gamma H2AX landing pad is called MDC1. That's mediator of DNA damage checkpoint protein 1.
MDC1 binds gamma H2AX.
Yep. And MDC1 then acts as a secondary scaffold, recruiting other factors, particularly E3 ubiquitin ligases like RNF8 and RNF168.
Ubiquitin ligases, so they start tagging other proteins.
They tag other histones, actually, adding ubiquitin chains. This further modifies the chromatin landscape around the break.
And that modified ubiquitinated chromatin is what brings in even more crucial effector proteins.
You got it. Key players like BRCA1 and 53BP1 are recruited to this complex, modified chromatin platform.
And they're super important, right? BRCA1 especially.
Critical, because they don't just help amplify the damage signal, they also play a major role in deciding which repair pathway gets used. Is it going to be a homologous recombination or non-homologous end joining? Gamma H2AX, through these downstream factors, helps make that choice.
Wow, so gamma H2AX is right at the epicenter of coordinating the whole repair response.
Absolutely. And from a clinical or research perspective, seeing gamma H2AX is really powerful, isn't it? Oh, hugely. Gamma H2AX foci, which are these visible clusters or dots of phosphorylated H2AX. You can see when you stain cells under a microscope, they're an incredibly sensitive biomarker for DSBs. They show up fast.
Super fast, within minutes. And the number of foci generally correlates pretty well with the number of DSBs the cell received, even after low doses of radiation.
And what if they stick around for a long time?
That's the key indicator. If you see persistent gamma H2AX foci hours or even days after radiation exposure, it's a strong signal that the cell is having trouble repairing the damage. It suggests ineffective repair and potentially radiosensitivity.
So it's like a direct visual readout of the damage and the cell's initial response happening in real time.
Exactly. You can actually see this in cells from patients with syndromes like ataxia-telangiectasia. Remember, their ATM is broken.
Right.
So their cells often show persistent gamma H2AX foci after radiation because they can't properly signal downstream to get the repair done efficiently. The signal gets stuck on. Okay, so ATM and ATR are activated. The gamma H2AX beacon is up. The scaffolding is being built. What's next in the signaling chain? What do ATM and ATR actually phosphorylate to really spread the alarm?
Right, so they're transducer kinases. They phosphorylate many downstream targets, hundreds potentially. But two of the most critical ones for cell cycle control are the checkpoint kinases we mentioned earlier, checkpoint kinase 1, CHK1, and checkpoint kinase 2, CHK2.
And this is where our earlier pairing comes back, right? The ATM CHK2 and ATR CHK1.
Spot on. Get this locked down. CHK1 is primarily activated by ATR in response to that replication stress or ssDNA damage. It's the major effector for the S phase and G2M checkpoints. Think of it as ATR's main lieutenant for checkpoint.
Okay, ATR CHK1 for ssDNA replication stress.
And CHK2 is primarily activated by ATM in response to DSBs. It contributes to G1S and G2M control, and it's also a key player in activating the critical tumor suppressor P53. So ATM's main checkpoint lieutenant.
ATM CHK2 for DSBs. This pairing seems really fundamental.
It is super high yield reminder. ATM signals mainly through CHK2. ATR signals mainly through CHK1. Burn that into your memory for the boards. These checkpoint kinases are the ones that actually relay the stop to the cell cycle machiner.
And that stop message leads us directly to cell cycle checkpoints. Why is pausing the cell cycle so crucial after DNA damage?
Because the absolute worst thing a cell with damaged DNA can do is try to divide before that damage is fixed. Imagine going into mitosis with broken chromosomes.
Yeah, that sounds like a disaster. Leads to aneuploidy, chromosome loss.
Exactly. It's usually lethal for the cell, or it leads to severely messed up daughter cells with massive genomic instability. So checkpoints buy the cell precious time to get the repair job done right.
Okay, walk us through the main checkpoints that get activated, especially after radiation damage. Let's start with G1S.
Right, the G1S checkpoint is crucial because it prevents cells that have damaged DNA before replication from entering S phase and trying to copy that damaged template.
Don't want to replicate errors.
Exactly. Here, ATM and its partner CHK2 are key players, especially after DSBs. One of their most important jobs is to phosphorylate and stabilize P53.
Ah, P53, the guardian of the genome. We keep coming back to it. Yeah.
You always do.
Yeah.
Stabilized P53 acts as a transcription factor. It turns on genes. And one of its most important target genes is the one that encodes P21.
P21, the CDK inhibitor.
That's the one. P21 is a potent CKI, a cyclin-dependent kinase inhibitor. It binds to and inactivates the cyclin E-CDK2 and cyclin D-CDK4/6 complex.
And those are the complexes that normally drive the cell into S phase.
Right. So if P21 inhibits them, the cell arrests in G1. It doesn't start replicating its DNA.
Okay, makes sense. What about during S phase itself? Is there a checkpoint there?
Yep, the intra-S phase checkpoint. This one primarily relies on the ATR CHK1 pathway because it's often responding to stalled replication fork.
Ah, the ssDNA signal again.
Right. If a replication fork hits a damaged spot on the DNA and stalls, the ATR CHK1 pathway gets activated. It does a couple of things. It slows down or halts the firing of new replication origins, and it stabilizes the stalled forks themselves, preventing them from collapsing. Basically, it puts the brakes on replication until the problem is fixed.
Okay, and then finally the G2M checkpoint. This one feels particularly important after radiation, preventing entry into mitosis.
It really is arguably the most critical one after radiation exposure. Preventing cells with unrepaired DSBs from entering M phase, mitosis, is absolutely vital.
And how does that work? Who controls the G2M stop sign?
Both pathways converge here. both CHK1, activated by ATR, and CHK2, activated by ATM, play roles. One of their key sets of targets are the CDC25 family of phosphatases, CDC25A, B, and C.
Phosphosis. They remove phosphate groups, right?
Exactly. CDC25s are normally needed to activate the main engine of mitosis, the complex called cyclin B-CDK1, also known as MPF, or mitosis promoting factor.
How do they activate it?
By removing specific inhibitory phosphate groups from CDK1. So CDC25 activity is required for CDK1 to become active and push the cell into mitosis.
So CHK1 and CHK2, they must inhibit CDC25.
Precisely. CHK1 and CHK2 phosphorylate the CDC25 proteins, and this phosphorylation leads to their inactivation or degradation.
So if CDC25s are inhibited,
then cyclin B-CDK1 stays inactive because those inhibitory phosphates don't get removed. And if cyclin B-CDK1 is inactive, the cell arrests in G2. It can't enter mitosis.
Clever mechanism. Does P53 play a role here too?
It can. P53 can also contribute to G2 arrest. One way is by inducing another protein called 1433 sigma. This protein can actually grab onto the cyclin B-CDK1 complex and sequester it in the cytoplasm, keeping it away from the nucleus where it needs to act to start mitosis.
Wow, multiple layers of control.
Absolutely. And it's important to remember, beyond just these checkpoints, this whole AT-MET gamma H2AX signaling cascade also phosphorylates and regulates many of the proteins directly involved in the actual repair pathways themselves, like NHEJ and homologous recombination. It helps recruit them, activate them, coordinate their actions right at the damage site.
So it's sensing, signaling, flagging the damage, stopping the cell cycle, and helping to activate the repair crews. It's a very integrated system.
Totally integrated.
Okay, so we've gone through the molecular details, sensing, signaling, checkpoints. This all sounds complex, but it has massive implications for our patients, right?
Yeah.
What happens clinically when these pathways are broken?
This is where it becomes incredibly clinically relevant and why you absolutely must know this stuff for the boards and for practice. Defects in these fundamental DDR pathways lead to severe genetic instability syndromes.
That's a classic example.
The classic example you have to know is ataxia-telangiectasia, or AT. It's caused by mutations in the ATM gene.
Okay, AT is caused by ATM mutations. What do we see in those patients?
Clinically, they typically present with cerebellar ataxia, that's problems with coordination and balance. They get telangiectasias, these small dilated blood vessels, often in the eyes and skin. They have immunodeficiency, and most relevant for us,
Extreme radiosensitivity.
profound, often life-threatening radiosensitivity. Even tiny diagnostic doses of radiation can sometimes cause severe reactions. Standard therapeutic doses can be absolutely devastating for their normal tissues.
Wow.
And because their cells can't maintain genomic stability due to the broken DSB response, they also have a dramatically increased lifetime risk of developing cancer, especially lymphomas and leukemias.
It really drives home how essential that ATM-mediated response to DSBs is for normal development and preventing cancer. If you can't sense and respond properly, you're incredibly vulnerable.
Exactly. And there are related syndromes, too. For instance, Nijmegen breakage syndrome, or NBS, that's caused by mutations in MBS1.
Part of the MRN complex, the recruiter for ATM.
You got it. An AT-like disorder, or ATLD, is caused by mutations in MRE11, also part of MRN. Both of these involve defects in those initial sensing components and also lead to varying degrees of radiosensitivity, immunodeficiency, and cancer predisposition. They're all part of this interconnected pathway.
These pathways also give us potential biomarkers, right? Things we can measure.
They certainly do. The status of genes in this pathway, like whether P53 is mutated in a tumor, or if BRCA1 or BRCA2 are mutated, remember they tie into the repair downstream, that can definitely affect prognosis and how a tumor might respond to treatment.
And gamma H2AX itself.
Yeah, as we mentioned, gamma H2AX foci are being heavily explored as a predictive biomarker.
How so?
Well, the idea is, if you could measure gamma H2AX foci in a patient's tumor cells, or maybe even their normal cells like lymphocytes, before or after a test dose of radiation,
You might get a sense of their repair capacity.
Exactly. If you irradiate cells and see gamma H2AX photos persist for a long, long time, it suggests poor repair capacity. That might predict increased normal tissue toxicity, meaning maybe you need to reduce the dose or change the plan. Or in the tumor, it might predict sensitivity, or sometimes, paradoxically, resistance depending on other factors. It's complex, but these assays are basically trying to give us a window into the cell's real-time repair status.
And maybe the most exciting part clinically is that this entire system is also a major target for new cancer therapies.
Oh, crucially so. This is a really hot area. Inhibitors that specifically target ATM, ATR, CHK1, and CHK2 are all actively in clinical development, many in trials right now.
What's the strategy there? Why inhibit the damage response?
The main idea often leverages a concept called synthetic lethality.
Okay, explain that.
So many cancer cells already have a defect in one DNA repair or DDR pathway. That's actually common in cancer, think BRCA mutations, which impair homologous recombination repair. These cells are often more reliant on the other pathways, like maybe the ATR pathway, to survive the DNA damage they constantly experience.
So they're already limping along on one leg, so to speak.
Exactly. So if you then come in with a drug that inhibits that other pathway, say an ATR inhibitor or a CHK1 inhibitor, you knock out their remaining crutch.
And that's lethal specifically to the cancer cell.
That's the hope. It's synthetically lethal because inhibiting either pathway alone might be okay, but inhibiting the second one in a cell that's already deficient in the first is catastrophic. Normal cells, which usually have both pathways intact, should tolerate the inhibitor much better.
So these inhibitors are designed to disable the cell cycle checkpoints, or essential repair steps, forcing those already unstable, damaged cancer cells over the edge into mitotic catastrophe or apoptosis.
Precisely. Or they can be used to sensitize cancer cells to traditional chemotherapy or radiation, making those treatments more effective by preventing the cancer cells from repairing the damage effectively. Understanding these molecular players really helps us understand these syndromes, potentially predict responses, and develop these really smart, targeted therapies.
Okay, fantastic overview. Let's try to distill some of this down into, say, 5 high-yield clinical pearls, things you absolutely need locked down for your boards.
Good idea. Pearl number 1. ATM is the primary sensor for double-strand breaks, DSBs, and primarily activates CHK2. Mutations in ATM cause ataxia-telangiectasia, AT, resulting in profound radiosensitivity. Remember, ATM-DSB, CHK2, AT, radiosensitivity.
Pearl number 2.
ATR senses single-stranded DNA, ssDNA, and stalled replication forks, and primarily activates CHK1. Remember, ATR, ssDNA replication stress NASH CHK1.
Pearl number 3.
The MRN complex, that's MR11, Rad50, and MBS1, is absolutely essential for detecting DSBs and then recruiting and activating ATM at the damage site. MRN finds the break, calls ATM.
Pearl number 4.
Gamma H2AX. It's phosphorylated H2AX at DSB sites. It forms those visible foci that are excellent biomarkers for DSBs, and it creates that critical platform or scaffold for recruiting other DDR proteins like MDC1, RNF8-168, BRC1, and 53BP1. It's your visual marker and recruitment hub.
And pearl number 5.
The key job of the checkpoint kinases, CHK1 and CHK2, is to block cell cycle progression. They do this mainly by phosphorylating and inactivating CDC25 phosphatases. No active CDC25 means no active cyclin BCDK1, which means G2M arrest. Also, remember ATM CHK2 can activate P53, which induces P21, which inhibits G1S CDKs for G1 arrest. Stop the cycle.
Perfect. Those 5 pearls really summarize the core connections. Okay, you ready for a quick board blitz? Let's see if you can apply some of this to typical board-style questions.
All right, let's do it. Hit me.
Question 1. A 5-year-old patient presents with cerebellar ataxia, oculocutaneous telangiectasias, and recurrent infections. Genetic testing reveals a homozygous mutation in the ATM gene. If this patient requires radiation therapy for a malignancy, which of the following is the most significant clinical concern? A. Extreme radioresistance requiring significantly higher doses. B. Increased risk of severe normal tissue toxicity due to radiosensitivity. C. Impaired nucleotide excision repair leading to increased UV sensitivity. Or D. A hyperactive G2M checkpoint causing prolonged cell cycle arrest.
Okay, ataxia telangiectasia's ATM mutation that screams ataxia telangiectasia. And hallmark of AT is radiosensitivity. So the answer has to be B, increased risk of severe normal tissue toxicity due to radiosensitivity.
Exactly right. Explanation. AT is caused by ATM mutations, leading to a defect in DSB sensing and signaling, resulting in extreme radiosensitivity. Classic association you need to know cold.
Got it. Next.
Okay, question 2. Which protein complex is primarily responsible for the initial detection of DNA double-strand breaks and the subsequent recruitment of ATM kinase to the damage site? A, the AAA complex, B, the MRN complex, C, the 9-1 complex, Rad9, Rad1, Has1, or D, the BRCA1, BARD1 complex.
Okay, initial detection of DSBs and recruitment of ATM, that's the job of the MRN complex, so B.
Correct again. B, the MRN complex. Explanation. The mRNA complex, mRE11, Rad50, MBS1, is the first responder to DSBs and is critical for bringing ATM to the site and helping activate it. Remember, ARRPA is for ATR recruitment to ssDNA. 911 helps activate ATR. BRCA1 comes later.
Makes sense. Keep them coming.
Question 3. Following exposure to ionizing radiation, the rapid phosphorylation of histone H2AX on serine-139, forming gamma H2AX, is considered a highly sensitive molecular marker primarily indicative of A, the initiation of basic scission repair, B, the presence of bulky DNA adducts, C, the induction of DNA double-strand breaks, or D, successful completion of DNA replication.
Gamma H2AX foci. That's the signal for double-strand break. So C, the induction of DNA double-strand break.
Nailed it. C is correct. Explanation. Gamma H2AX foci form rapidly and specifically at sites of DSBs, making them a widely used and very sensitive marker for these critical lesions induced by radiation.
Right, one more.
Last one. Question 4. The ATR kinase pathway is primarily activated by replication stress and single-stranded DNA. A key downstream effector kinase phosphorylated and activated by ATR, which subsequently targets proteins like CDC25A for inhibition to enforce cell cycle checkpoints, is A, CHK2, B, ATMC, P53, or D, CHK1.
Okay. ATR pathway, ssDNA replication stress, downstream kinase inhibiting CDC25. That's got to be CHK1. ATR activates CHK1, so D.
Perfect. The correct answer is D, CHK1. Explanation. ATR is the primary activator of CHK1 in response to replication stress. Activated CHK1 then plays a critical role in the S phase and G2M checkpoints, partly by phosphorylating and inhibiting CDC25 phosphatases. Remember that key pairing, ATR-CHK1.
Got it. That blitz helps solidify the connection.
Excellent job. You see how breaking it down and focusing on those core players and their main functions and connections, like ATM goes with CHK2, ATR goes with CHK1, mRN recruits ATM, gamma H2AX marks the DSBs, and the checkpoints block CDC25 or activate P21. That's how you tackle these questions.
Absolutely. And this isn't just, theoretical basic science. This whole area of DDR signaling is a huge area of ongoing research and clinical translation. It's moving fast.
Right. As we mentioned, these gamma H2AX assays are being explored more and more rigorously for potentially predicting individual patient or tumor response to radiation.
And the development of those inhibitors targeting ATM, ATR, CHK1, and CHK2 is really exciting, especially for potentially using that synthetic lethality approach in cancers that already have repair defects like BRCA mutation.
Yeah, understanding these pathways truly helps us understand why some patients are just inherently radiosensitive because of germline mutations, like in AT.
And it gives us a roadmap for how we might personalize therapy much more effectively in the future. Based on a tumor's specific molecular profile, its specific DDR defects, this really is the foundation of precision radiation oncology.
Okay, so to quickly pull it all together one last time, summarizing our look into DNA damage, sensing, and signaling.
Radiation-induced DSBs. Primarily sensed by the ATM pathway, which gets kicked off by the mRN complex, mR11, Rad50, NBS1.
Replication stress and single-stranded DNA. That activates the ATR pathway, working closely with ATRPA.
A core early signal right after DSBs is gamma H2AX formation, that's phosphorylated H2AX. It marks the break visually and builds a scaffold for recruiting key repair and signaling proteins.
Activated ATM and ATR then act as signal transducers. They activate the checkpoint kinases, primarily CHK2 downstream of ATM and CHK1 downstream of ATR. Remember that pairing.
Right. And these checkpoint kinases, along with P53, orchestrate cell cycle arrest to allow time for repair. That means G1S arrest via P53 inducing P21, which inhibits cyclin CDK.
And G2M arrest, mainly by CHK1 and CHK2, inhibiting the CDC25 phosphatases, which prevents cyclin BCDK1 activation. Stop the cell cycle.
And finally, remember that defects in these fundamental pathways cause those severe radiosensitivity syndromes like ataxia-telangiectasia, and these pathways are also prime targets for developing new, smarter cancer therapies.
Mastering these core components, their connections, and their clinical relevance is absolutely essential for crushing your RadBio boards. It seems complex, but you can definitely get it. Keep reviewing these pathways, maybe draw them out, visualize that molecular dance, and keep connecting the dots between the molecules and the clinical picture. You've got this.
Absolutely. Keep putting in the work. It pays off.
That wraps up our discussion today on DNA damage sensing and signaling, a really crucial topic.
For sure. And for more practice with questions like the ones we just did, and to test your understanding even further, You can find practice oral boards and other great review materials over at radoncsmartlearn.com.
And definitely subscribe to Radiation Oncology Smart Review for our next session. We'll be diving into the cell's repair machinery itself, focusing on the two major DSB repair pathways, non-homologous end joining NHEJ and homologous recombination repair HR.
Yeah, I'm going to talk about how the cell actually fixes those breaks. Thanks for tuning in, everyone.